Well, folks, welcome to Healthcare Unfiltered. I'm excited today because I have a newcomer. It's the first time that Dr. Gavin MacBeath joins me on Healthcare Unfiltered. So, Gavin, welcome to the show. I appreciate you taking the time for joining me.
Yeah, thank you for having me.
It's not easy always to find time on a busy schedule. You are the CEO of a company called TScan, a biotech company. We're going to talk about that. I'm fully aware and cognizant of how challenging it is to find time for these. So it does not go unnoticed that you found time for my podcast.
Well, I will reveal that this is actually the first podcast I've ever done. So, looking forward to the experience.
So I'm going to say this is a podcast exclusive. There you go. It qualifies. Um, Gavin, tell us a bit about you. I mean, not the CEO part or the leader of TScan, more about the person behind the executive.
Sure. Yeah. So, I actually was born in South Africa, spent some time living in Thailand for a bit. But ultimately, my parents emigrated to Canada when I was about 7 years old. And so grew up in Canada, in Winnipeg, Canada. And then, growing up there, got very much interested in going into the medical field and healthcare in general. So did my undergrad degree in Winnipeg at University of Manitoba, and then moved down to the United States for grad school in San Diego, and then ultimately moved up to Boston, where I came for postdoc studies at Harvard, and loved the Northeast. Loved growing up in Boston. Raised my family here. Have three kids that are now all young adults. And so ultimately started my career in academia. I was actually professor in the chemistry department at Harvard for about 7 years, and spent my time really working on trying to understand cancer, understanding cancer cell at a systems level. And so while I was at Harvard, I co-founded a company called Merrimack Pharmaceuticals, and so made the decision back in 2008 to leave academia and move full-time to industry. Felt this was the greatest opportunity to really have an impact on patient lives by developing therapeutic products. And so worked at at Merrimack for about 7 years. Ultimately, Merrimack was successful in getting a drug approved for pancreatic cancer, and proud to say that Onivide is now part of frontline therapy in pancreatic cancer. But you know, while I was at Merrimack, there was a whole revolution in cancer therapy. The whole field of immunotherapy really blossomed. And so I just came to recognize the importance of the immune system in fighting cancer. And so when the opportunity came up in 2018 to get in at the ground floor of a company developing novel T cell therapies for cancer, sort of jumped on the opportunity. And so started initially as the chief scientific officer of TScan, and then about 2 years ago, moved into the role as CEO of TScan.
You're a scientist by nature, so you're in the right spot to do this. What was so different between being the scientist on the academic side at Harvard and all of that versus the scientist, but now you're on the manufacturing side or on the biotech side?
Yeah, so being at Harvard is, really enriching experience for me. Did a, time is really focused a lot on research, and for me, it was a lot in basic research. So really understanding, the inner workings of the cell and how cells signal, receive, and process signals. So I'd say it was a real opportunity um, to really do sort of groundbreaking research at a fundamental level on basic biology. But I really felt that it was too far for me connected from actually affecting patients' lives. Whereas in biotech, you're working directly on something that has a direct connection to human health, right? So while I was at Harvard, people would ask me, "What are you working on?" And the problem is, it would take 5 to 10 minutes for me to explain what I was working on. And then they would inevitably ask, well, how is this going to translate into helping people? And it's, well, one day these fundamental discoveries may or may not, come into reality and affect patients' lives. Whereas in biotech, I just find it personally to be a very rewarding experience because I can directly connect what I'm doing with ultimately having an impact, in healthcare. And so now the short answer to the question, what do you work on, is I'm developing therapies for cancer.
Yeah, yeah. You mentioned before you joined TScan, you formed a company, Merrimack, and I'm actually familiar with the company and with the drug. How difficult is to form a company that you really want to take it from an idea to form something that eventually, gets acquired or sold, but eventually develops something that is really get the, I mean, how do you get there? How challenging is this?
Yeah, so for me, I didn't set out to form a company when Merrimack began. At the time, I was working on a new technology in my lab at Harvard. It was a way to understand signaling networks at a systems level. I was working on protein microarray technology. And I was approached by some folks at MIT who were themselves also working on, understanding cells at a systems level, but with a goal of starting a company. And so for me, it was an opportunity to learn what company formation was about without having to do it all myself, right? So as part of the, one of 5 or 6 founders of the company, everyone came with different levels of experience. I was bringing the technology that became the basis for the early work at Merrimack, but others were bringing experience in founding companies and acquiring funding. And so the whole process for me was a learning experience because it was setting up the initial science, but also from the business side of things. Merrimack made a decision in the first year of the company to merge with another company called Atlantic Biopharmaceuticals, and that sort of brought together the full team that really carried Merrimack through those early years. And then I also learned that companies evolve over time, right? What you start with as your founding technology or your founding idea doesn't necessarily translate into what becomes the ultimate product. And so ultimately, Onivide came through a merger that we did, or an acquisition that we did at Merrimack, of another company, a West Coast-based company that brought this lipid nanoparticle technology to the company that ultimately was the basis for Onivide. So the whole process for me was just, a real learning experience in how you take early stage discovery, develop it into a pipeline, but also bring the business side of things through strategic acquisitions or strategic mergers to ultimately put together a company that can bring a drug to market.
In your current role, you're the Chief Executive Officer. What does your day look like? How do you spend your day? And are there opportunities for you to wind down sometimes?
Yeah, so I come at the CEO position from that background in science, right? So for me, a lot of my day is actually understanding what's going on, at fairly high detail in terms of the various preclinical and clinical programs going on at the company. So I do spend a lot of my time in meetings internally, meeting with the preclinical team, meeting with the clinical team, meeting with our manufacturing group to really understand what are the challenges that they're facing on a day-to-day basis. I do some problem solving in there, but really just kind of guiding and steering the strategy of the company as we go through the day. I also spend a fair amount of my time meeting with investors, doing the typical investor relations work, keeping people apprised as to what we're doing. So it's a bit of a balance between sort of the internal focused side of the job of working with the teams at TScan to stay on track with our overall goals as a company, and then also connecting with the investment community.
So in preparation for this, Gavin, I was reading a lot about the company, about what TScan does, and on your website, there's a lot of information about the pipeline as well as about what the company does. But at its core, at least what I gathered from reading, is that you are focused on T cells. Basically, as a company, you are leveraging T cells to fight cancer. Tell us what that means. Like, what exactly does that mean?
Sure. So, by far the best defense against cancer is the human immune system, right? So, the human immune system has evolved to fight off external threats, right? So, pathogens, but it's also evolved to fight off anything that is not normal. And cancer is something gone wrong with your cells. And so, it's actually T cells is the component of your immune system that most actively fights cancer. So essentially, T cells are this component of your immune system that's constantly surveilling the body to see if there are any cells that something's gone wrong with the cell. And when they see that something's gone wrong with the cell, their job is to eliminate that cell. And so because of that, I think that T cells are the best therapeutic option for us as well. And this is actually borne out over the last 10 to 15 years in the clinic. So where this whole revolution in immunotherapy started was with what are called checkpoint inhibitors. And what checkpoint inhibitors do is they essentially unleash your T cells. So T cells have this natural mechanism of being shut down, and cancer cells actually co-opt that system to shut down T cells. And what checkpoint inhibitors do is they basically unleash those T cells to act and to do what they're supposed to do. So that was sort of the first wave of immunotherapies, is unleashing a patient's own T cells. But then the next wave of immunotherapy was in learning to genetically reprogram T cells. So even though checkpoint inhibitors work, they don't work for all patients because you don't necessarily have T cells that are geared to fighting your cancer. But if you can genetically reprogram a patient's T cells, then you can direct them towards fighting their cancer. And so with the advent of what are called CAR T cells, CAR T therapy, it was a way of genetically reprogramming a patient's T cells. And that's proven really effective for certain types of cancer. But ultimately, what we're doing at TScan is using the natural receptor of the T cell, called the T cell receptor. We're using the natural mechanism of T cells to reprogram a patient's T cells to recognize and fight their cancer. So that's the basis of what we're doing at the company, using naturally occurring T cell receptors to genetically reprogram patients' T cells to recognize and then fight their cancer.
So you're using the receptor to modify the T cells.
Yeah, so essentially find a T cell receptor that recognizes a cancer-specific antigen. So this is an antigen that a cancer cell is making that normal cells don't make. So, for example, if your cancer results from certain mutations occurring, that's not normal to the cell. The T cell would recognize that as being abnormal and fight the cell that has that mutation. So if you can find a T cell receptor that recognizes that antigen, then you can take a patient's T cells. So basically from a process called apheresis, where you take a patient's T cells basically through a prolonged blood draw. We then get those T cells at the company and in a clean room, add a gene that introduces that T cell receptor into the T cells, grow them up to large quantities, and then send them back to the clinical site where they get reinfused through IV infusion back into the patient. Those T cells have now been reprogrammed to recognize that cancer-specific antigen, so they'll only go after the cancer cells in the patient, but not touch any of their normal cells. So essentially a way to reprogram a patient's natural T cells to fight their cancer.
The process, though, the way you're describing it in terms of the leukapheresis, expansion, reinfusion, it's like what we do with CAR T, no? I mean,
yeah, so it's basically exactly the same process as CAR T, except rather than using, so what the CAR stands for in CAR T is chimeric antigen receptor. Instead of using a chimeric antigen receptor, we're using the natural T cell receptor. So basically CAR T therapy, but using T cell receptors rather than chimeric antigen receptors.
So scientifically, if you put your scientific hat now, is how much of that is a differentiator where you think does this give an edge when you're using the natural T cell versus the other CAR T, or is it just a different methodology? Does it have a scientific edge or?
Yeah, so it does have a scientific edge. So there's really two reasons why we're doing this. So for a start, CAR T receptors, CAR Ts, they can only recognize cell surface proteins. So there's actually a limited number of cancer-specific cell surface proteins available to target. Whereas T cell receptors, they can recognize not just cell surface proteins, but also intracellular proteins. And so that just opens up a much broader set of targets that you can address with TCRs or T cell receptors that you can't address with CARs. So that's the first advantage, is it just opens up a large swath of targets that you can go after. And then the second advantage is that we're taking advantage of literally 500 million years of evolution, right? So, we as humans have evolved to have an immune system. And so that T cell receptor has been perfectly tuned over time to signal the cells appropriately, not to oversignal them, not to undersignal them, but to provide the right signals to the T cell to cause them to get activated, to recognize the cancer, to kill the cancer, to form memory cells. All of these are unique properties to T cells that have been, evolved over hundreds of millions of years. And so that's what you're really taking advantage of when you use naturally occurring T cell receptors as opposed to CAR T. So in that long answer, I did include the fact that T cell receptor engineered T cells can form memory cells as well. And so part of the reason that you can respond faster to a disease that you've had previously is that your T cells have formed memory cells that can jump into action again. In fact, that's the basis for how vaccines work. You're pre-priming your T cells to recognize a pathogen so that years later, if that ever comes up again, your T cells can jump into action quickly and attack that that pathogen. And so the same thing with these cancer products, right? Once we form those TCR-T cells, they can form memory cells. And if the cancer ever comes back, they're in position to go and fight that cancer again. So that's the other advantage that TCR-T cells offer relative to CAR T cells.
It's very helpful, Gavin. Are you focused on particular diseases to apply this technology?
Yeah, so TScan, we've got two main programs, one in heme malignancies. So this includes AML, ALL, and MDS, or sort of very common, heme cancers. And so that's our lead clinical program. And in fact, we're gearing up to start a phase 3 trial on that by the end of the year. And then we also have a program in solid tumors. And so in solid tumors, we're focused on cancers like non-small cell lung cancer, head and neck cancer, and various forms of sarcoma, all of which have clear unmet needs, and we're desperately in need of novel immunotherapies for second-line therapies and subsequent therapies. So the lead program is the heme program in AML, ALL, and MDS, but we're following that up with our solid tumor program as well.
So I guess my follow-up question to that is why these diseases are there? Is it more of a strategy, or is it actually more that these are the diseases that are more likely to benefit from this technology using TCR that you just described?
I think it's a little bit of both. So when we started the company, the big goal was to develop therapies in solid tumors, and particularly to introduce what we call multiplex therapy. So treating a patient with more than one T cell product at a time. And there, it's really to address the fact that solid tumors, one of the biggest challenges is that they're heterogeneous. So not every tumor cell is the same. And so having sort of more of a natural response. So in nature, you don't just make one T cell against cancer, you make multiple different T cell clones. Similarly, coming in with a product that's a mixture of many different T cell receptor engineered T cells is a much better chance at addressing solid tumors. So when we started the company, I really wanted to develop this, what we call multiplex therapy for solid tumors. But that's a very long path to get there. And so in the early years, we started initially working on solid tumors, but then we saw this opportunity in heme malignancies where there's a clear unmet need in AML, MDS, and ALL for patients that are naturally undergoing bone marrow transplants. And so bone marrow transplants are very effective. In fact, many people are cured by a bone marrow transplant. But unfortunately, if you relapse, then the prognosis is very poor. So 80% of patients will die within 2 years of relapsing if they have a bone marrow transplant. And there was this well-established literature around how T cells can prevent relapse in patients undergoing transplant therapy if you can direct them to certain types of antigens. And so early on in the company, as we're working on this sort of broader solid tumor program, we saw this real opportunity to work in the area of bone marrow transplants and develop a therapy that would prevent patients from relapsing when they undergo transplant. And so that program progressed at a faster rate than the solid tumor program. We got that into the clinic earlier, and we now have very compelling data in that heme program that, at least early stages, and this is not, phase 3 data or anything, this is from our phase 1 trial, but it appears that we're having a really substantial impact on patients undergoing bone marrow transplants. And so that's the program that's become the lead program for the company because of this early compelling data from our phase 1 study. So that's why we're gearing up to start a phase 3 study this year, even while we're moving the solid tumor program forward.
So historically, we've always been told that for solid tumors, the reason sometimes CAR Ts don't work, or I guess the research is a bit premature, is because it's very difficult to find that cell in a solid tumor that has a specific receptor that you can actually modify, and which is very different than in heme malignancies. In ALL, you have the CD19, for example, and you can do a CAR and all of that. Is your technology able to overcome that challenge when it comes to solid tumor? You mentioned lung cancer, sarcoma. Is that a far-fetched thought, or is the technology able to overcome that?
Yeah, so that's not a far-fetched thought at all. In fact, that's really the advantage of using TCRs rather than CARs. So, as I said, one of the advantages of the approach that we're taking is that there are far larger universe of targets that you can address with TCRs relative to CARs. And so I think one of the challenges that CARs have had in solid tumors is that there just aren't a lot of good targets to go after. You really need the target to be cancer specific and not to be expressed in any normal cells. Whereas with TCRs, because you can address these intracellular proteins, the universe is much larger in terms of what you can address. And there turns out to be a fair number of different proteins that are very cancer specific that you can address with TCR-T. And so we're developing a whole suite of products that we can give combinations of in our solid tumor program. So we actually have 7 different TCR-Ts in clinical trials right now in the solid tumor program. And that whole program is designed so that we can mix and match them so that we can give patients combinations of these 7 products, which we think, is the better solution for solid tumors to be able to address that heterogeneity.
What are the challenges as you build this program? And the reason I ask is because it takes a long time to get even the answer. I mean, you're just about to start a phase 3 study, like you said, and I don't know what you're thinking, but it could take 5 years to read. It could take 2 years to read. It depends on a lot of variables. And the field is moving during those 2, 3 years. I guess how do you build something and you mitigate the risk that by the time you even have the technology ready, it's not obsolete? And people say, well, it's great, but we moved on. Now we're doing something different.
That is a great question and probably the eternal question in biotech. Right? So for us, this heme program, we've actually got a trial designed that we believe can give us a readout that could potentially support full approval in 2 years. So if we launch that study this year, the goal is that by the end of 2027, we would have the top line data necessary to file for approval. It turns out that in this particular setting that we're operating in, right, patients undergoing transplant therapy, there's currently no other good treatment options. There's nothing else approved in the area, and there's very few clinical trials that are underway specifically to prevent relapse in patients undergoing transplant. So it's sort of the ideal place to begin a program where there's no direct competition, where you've got a very targeted patient population. You know where those patients are because they're all in transplant centers undergoing transplant therapy. So everything sort of lines up well for having that as a successful clinical and commercial program. In solid tumors, right, the bar is constantly changing, the target's moving. And so there you have to be very strategic to identify an area that clearly has an unmet need, where you don't see any immediate therapies coming up that you think could potentially supplant it. But our goal ultimately as a community is to come up with better and better therapies. And so from a non-CEO hat, but from a medical community hat, we want to continually find better therapies that supplant older therapies, right? As the field continues to develop, it'd be great to get better and better therapies for cancer. But strategically, obviously, focus on the areas with greatest unmet need, where there's no sort of direct other trials going on that could supplant what you're working on.
What has been your biggest challenge? I mean, since you've assumed the role of chief executive officer of the company, I mean, are there anything that you did not expect that you faced? Although, I mean, you were the CSO, obviously, so you had a lot of exposure into what's going on, but it's still different than being the CEO.
Yeah, I mean, I would say, sort of the biggest challenge has been that biotech is obviously in a very challenging period of time right now. So obviously, just with where the economy lies, with where even interest rates affect biotech investment, a lot of factors come into play, sort of uncertainty around regulators at this point. All of these things have sort of aligned to make biotech a particularly challenging place to be operating right now. And so I would say, one, being able to present a very clear vision for where the company's going. Two, having really supportive investors. That's been critical to TScan's success. We have a sort of core group of investors that have remained committed to the company and very much aligned with our vision. So that's really been the challenge, is to kind of navigate the current sort of challenging funding environment, to keep the company on track, to keep it well funded, and to really try to get to the point of getting an answer on the scientific hypotheses that we built the company around. So that's I would say been the biggest surprise and biggest challenge since taking on the role as CEO, is keeping the company on track strategically, but also keeping it sort of well capitalized so that we can get to those key data readouts that then support the next round of funding for the company.
A while back, Gavin, I was interviewing on my podcast a CEO also of probably a smaller company. It was more like in the startup stage. And I asked her actually the same question. And what she told me was interesting, and I'd like her to react to it. She said that one of the challenges that she actually didn't appreciate before was sometimes investors, they want to have a lot to say into what you need to do internally and the direction and even the scientific piece and so on. And while I understand the concept, because if you are the investor and you're keeping the money, the company afloat, you want to have a lot of say in it. But they're not usually scientists. They're not usually physicians or PhDs and so on. So one of the things she shared with me that sometimes communicating that piece to investors and explaining to them why maybe their idea is not the best idea is not always the easiest conversation to have. I mentioned that because I wasn't sure if this is something that you have to deal with or not, but I have heard that from some CEOs to kind of keep the investors understanding the science enough, but sometimes they want to change everything if they don't really like what they see, and you have to keep them in check.
Uh, that's very good points. I would say we've just been particularly privileged as a company. So let me give you a little bit of a history of TScan. So the company was founded in 2018, and there was initially a consortium of 5 different venture capital funds that put in the seed round to start TScan. And the company was founded on a technology that enables us to discover new targets for TCR therapy. So the early years, we were studying tumors from cancer patients to try and discover new targets and develop these therapies. But very quickly, we developed our clinical programs and got going into clinical trials early on. But one of the sort of transformative moments for the company was when we did our Series C financing, which was in preparation for an IPO. And we got a whole new set of sort of public-facing investors. And in particular, one investment group, Baker Brothers, came in and led that round, and have been very supportive of the company throughout its subsequent development. And so it turned out that when a company has an IPO, typically all of the investors that were representing the VCs, they, according to their own rules, exit the company at that point. And so since our IPO, we've only had one investor representation on the board, right? And that's, from the lead investor, from Baker Brothers. And I think having just one, investor on the board and the rest be independent board members has made things very smooth as a company, because you don't have this sort of conflicting views from different investors. And the other thing is, the Baker Brothers are incredibly intelligent individuals, right? And very well versed. And so we haven't had major disagreements in terms of where to take the company. We've been very aligned. And so I just have been fortunate in terms of not having to deal with conflicting views on the board, or conflicting views of where to take things, because at least so far we've been very well aligned. But I understand that is a key challenge in the business. We just happen to be in a very fortunate situation as a company.
So looking ahead, Gavin, like the next couple of years, you're focused on these pivotal studies, it seems like, and the readout and where this would take you. Is that a fair assessment? That's really the short term, what you're focused on?
Yeah, so in the short term, launching this pivotal trial, and then advancing the solid tumor program to see if we're actually having the impact that we, set out to have, whether this concept of multiplex therapy is actually going to bear out in the clinic. So I'd say, by the end of this year, launching the pivotal trial is sort of the top priority for the company. And then second, generating the data in solid tumors that enable us to sort of make key decisions as a company as to where to take that program.
I ask every CEO and every leader that comes on my show the same question. I'm going to ask it to you as well, which is, if people look from the outside, how would they describe your leadership style?
So I would say tending towards the more consensus building, right? Less rather than top down. So I have a very talented group of senior leaders at the company, C-suite, SVP, VP level leaders. And so we tend to work very collaboratively, meet quite regularly. But I seek input from all the different parts of the company before making any decision. So it's not a democracy, but it's very much a sort of a cooperative process in terms of getting all the information necessary to make these key strategic decisions. So I would say consensus building style of leadership.
How big is the company now, Gavin?
We're about 200 employees.
Hmm. Yeah.
And big part of that is that we do all our own manufacturing internally at the company. So we have our own GMP clean room where we do the cell therapy manufacturing. We have our own QC, QA, as well as all the discovery, preclinical. Everything we put into the clinic has actually come out of our own discovery program. Nothing's been in licensed at this point. So it's sort of this fully integrated biotech company.
Any final thoughts? Any things I should have asked you that I did not ask you? First of all, congratulations. I really love what you're doing, and I think it's always fun to see ideas translate into actual products, and that hopefully this will lead to patients being served. But any other thoughts, things I may have forgot to ask you before I let you go?
Um, Nothing springs to mind. I will say that one of the things that's driven me from the beginning of the company is that when I left Merrimack and was looking for my next opportunity, what I set out to do is I really wanted to follow the entire process from inception of an idea through to commercialization of a product. And so it's been a really rewarding experience since starting in December 2018. Coming up with the initial clinical programs, including the heme program, discovering all the products internally, building the manufacturing group, building the preclinical and now clinical group. This is sort of a dream, right? To be able to go from coming up with an idea and following a product all the way through to hopefully commercialization. So schedule me for your show
Well,
2.5 years from now.
I'll be rooting for you, and really
And hopefully in
congratulations
two and
on
a half years, I'll be able to say, hey, we're, getting ready to commercialize our first cell therapy product.
as they say, it's a journey. Well, congratulations on everything so far. Best of luck as you continue to progress forward. And thank you for coming on Healthcare Unfiltered. Dr. Gavin MacBeath, thank you so much for coming on.
Yeah, thank you again for having me. It was a pleasure.